Welcome to the BioInsights, where science comes alive.
Deep inside a microscopic community, bacteria are building something far more powerful than individual cells: a biofilm.
A biofilm is a structured community of microorganisms attached to a surface and surrounded by a protective extracellular matrix.
What begins as individual bacterial cells can develop into a highly organized and persistent microbial community.
The bacteria become embedded in an extracellular matrix that acts like a protective shield around the community.
Think of a biofilm as a microscopic fortress, where bacteria are protected behind a complex extracellular barrier. When antibiotics attack, the biofilm environment can make it much harder for treatment to reach and eliminate bacterial cells. Some bacteria can survive within these communities, contributing to persistent and difficult-to-treat infections. This problem becomes even more serious as antimicrobial resistance continues to threaten effective treatment of bacterial infections. When conventional antibiotics become less effective, researchers need alternative and complementary strategies. One fascinating possibility comes from nature itself: viruses that infect bacteria, known as bacteriophages. Bacteriophages, or phages, are viruses specialized to recognize and infect bacterial cells. A phage must recognize compatible features on its bacterial target before infection can begin. Once the correct bacterial surface is recognized, the phage attaches to the cell. The phage then delivers its genetic material into the bacterial cell, beginning its infection cycle. But attacking bacteria inside a biofilm is different. The phages must navigate the complex microbial matrix. The extracellular matrix can influence how easily phages move through the biofilm and reach their bacterial targets. Interestingly, phages bring another potential weapon to the fight: specialized enzymes such as endolysins and depolymerases. Depolymerases can target certain polymeric components of the biofilm matrix, potentially helping break down its protective structure. By disrupting parts of the matrix, these enzymes may help expose bacteria that were previously shielded. Endolysins are another important phage-associated enzyme group, known for targeting bacterial cell wall structures. By damaging susceptible cell wall structures, endolysins can contribute to bacterial cell destruction. Now imagine combining these activities. One mechanism attacks the bacterial fortress, while another attacks the bacteria themselves. The result can be a more accessible biofilm environment, potentially making bacterial cells easier to target. But bacteria are diverse, and a single phage may not effectively target every bacterial population. That is why researchers can investigate phage cocktails containing multiple phages with complementary target ranges.
Together, different phages may provide broader coverage and create multiple pressures against a complex bacterial community.
And researchers are also exploring another strategy: combining phages with conventional antibiotics. Combining phages with conventional antibiotics. This is called phage-antibiotic synergy, where the two approaches may enhance bacterial targeting in complementary ways.
Instead of relying on one weapon, scientists are investigating coordinated attacks against both the biofilm and its bacterial inhabitants. But bacteria are not defenseless. They have evolved multiple strategies to resist viral attacks.
Bacteria can alter or lose phage receptors, making it harder for particular phages to recognize and infect them.
They can also use intracellular defense mechanisms that recognize or interfere with invading phage genetic material.
This creates an evolutionary arms race, with bacteria and phages continually adapting to one another. A mature biofilm is not simply a pile of bacteria. It is a complex environment with different cells, conditions, and protective structures.
That complexity means successful treatment may require more than one mechanism of attack.
The emerging philosophy is simple. Instead of one weapon, use complementary strategies that attack different vulnerabilities.
Future approaches may increasingly depend on matching effective phages to specific bacterial targets.
Researchers are also investigating how phage-derived enzymes might be optimized for more effective biofilm disruption. The potential applications extend across medical and industrial environments where persistent biofilms create serious challenges. The fight against biofilms is becoming a battle of multiple strategies: viruses, enzymes, antibiotics, and scientific innovation.
Phages and their enzymes are promising tools, but important questions about effectiveness, resistance, delivery, and safety remain.
The future may belong not to a single miracle treatment, but to carefully designed combinations targeting biofilms from multiple directions. From viruses that hunt bacteria to enzymes that dismantle their defenses, science is discovering new ways to fight the biofilm fortress. This is the BioInsight. Subscribe and keep exploring the science behind microbes, biotechnology, and the future of medicine.
Thanks for watching the BioInsights. Like, subscribe, and follow for more.